Okay, so this is recitation week 12. I wanted to let everybody know that there's only one quiz left, and there's also only one homework left after this one this week. And next week is the second to the last time that I will be seeing all of you. So just to start it off, this recitation is kind of heavy. There's a lot of material in this, so we're gonna push our way through. And luckily, since this is recorded, you can skip through to the places you need to and watch them again. So starting off, the absence of available O2 limits oxidation of glucose, so cells in anaerobic conditions depend on glycolysis. Since the net gain is 2 molecules of ATP for glucose, the conversion is only really like 3% of available energy used for cellular use. It's going to convert pyruvate into lactate or ethanol. Those are only under anaerobic conditions. This is a review from last lecture. The last recitation, I ran out of time to go over it, but pyruvate is reduced into lactate. That's why our muscles start to burn when we work out too hard. And pyruvate is reduced to ethanol, is the alcohol that we drink. You can see here, there's two different pathways that you can go through. You can go through the lactate pathway or the ethanol pathway. The lactate pathway is going to use different enzymes. It's going to use lactate dehydrogenase. And then ethanol is going to be using acetaldehyde. It's going to have acetaldehyde as an intermediate, but it is going to be using pyruvate decarboxylase and alcohol dehydrogenase. You can see those different enzymes that are used here. Oh, I forgot to... Oh, wait. Present. Wait, no. Okay. But with the presence of O2, it allows for more things that you can do, which is aerobic respiration, which we are going to be going over in this recitation. As it goes on through aerobic respiration, more ATP is made from glycolysis. So, glycolysis and acetyl-CoA synthesis. With O2, you can make glucose. You can turn glucose all the way to H2O and CO2. You can get many more ATP out of one glucose molecule rather than getting only 2 ATP from one glucose molecule. The first step is to convert pyruvate into acetyl-CoA. Acetyl-CoA is the basis for generating many of the ATP biosynthesis. So there's acetyl-CoA synthesis, glycolysis, pyruvate, and it turns to acetyl-CoA right here, which we'll be going over. Wait, I want to go back. Okay. So you can see that the electrons are being pulled off here. That's why NAD comes in, and then you can see that pyruvate is kind of similar to acetyl-CoA, except that it has the coenzyme A group, and it took off some groups there. You don't really need to know what is being done, I say. So mainly you just need to know this part, evolution of cellular respiration. So you start from the cytoplasm. These are different locations that it's happening in, and the conditions it can happen in. So it can happen in anaerobic conditions. This can happen in atmospheric conditions, aerobic conditions. These can only happen with O2 present. So you have glycolysis, where you have starting molecule is glucose. There's phase 1, phase 2, phase 3, and then the end product is pyruvate, and then you have the production of acetyl-CoA. The starting product is pyruvate and makes acetyl-CoA, and then that acetyl-CoA is going to feed into the citric acid cycle and the electron transport chain. Right here, it shows you that there's a modest yield of ATP, only 2 of them are made, and the reducing coenzyme carriers are NAD+. And then here, when oxygen is involved, there's generation of many electrons, which you're going to be using NAD+, and then using those electrons to generate many ATP to restore generate many ATP, and you're also going to be using oxygen at the final step to restore the carriers, which we'll be going over. So this right here, we're comparing the 2 different methods of making ATP. Glycolysis alone involves limited oxidation of glucose, depending on enzymes transferring phosphates from a substrate to ADP and another phosphate group. So there is substrate level phosphorylation, which is right here. You're going to be using enzymes to transfer phosphates from a donor molecule to an ADP. So you can see the ADP, and it adds another phosphate on there to make it into an ATP. A pro of this is that it works, but it's not very efficient, so the cell doesn't utilize this as much. However, we have to keep it there because it's like evolution. So this is used in certain steps of phosphatative oh my god. It's going to be used in multiple steps of making ATP. I'm twisting my words right now. But this is the more efficient process. You're going to be using proton pumps and membrane channels. So the process is called chemoosmosis because using the protons to flow through the membrane channels, like osmosis sorta, and it makes ATP. It's like harnessing crowds pushing through turnstiles. So it's like these people pushing through these, and you're using that energy that's created from people pushing through all of those turnstiles in the subway station. It's very efficient. It makes tons of ATP, and we like it, but it requires a steady source of electrons to power that, and that's why throughout all these steps, we're going to be taking electrons off to feed into this to make ATP. So you can see here, F-type ATPases. F is for factor, also called ATP synthesis. You don't really need to know like F-type ATPases much. You just need to know it's in the inner mitochondrial membrane, right here, plasma membrane, and thylakoid membrane. So thylakoid membrane is going to be in the plants, plasma membrane is in bacteria, inner mitochondrial membrane is for us. And they're all using the proton gradient to drive ATP synthesis. So you can see all of those pumps are going to be located along here, the membrane, inner membrane. You should know the structure of a mitochondria because you're going to be asked a lot of questions like, oh, where are protons going to flow from, and like, are protons located in the outer membrane, inner membrane, are they located in the matrix? And you need to know where the structure of your mitochondria so you can identify and answer that question. So you can see here, this is the cytosol of the cell, is where glycolysis happens, and pyruvate oxidation also happens right here. Well, no, it's going to make the pyruvate, and then the pyruvate is sent into the mitochondria, and pyruvate oxidation is going to happen right here, and it's going to turn into acetyl-CoA, and then the citric acid cycle, and then the the electron transport chain. And one thing to notice is that you see the inner membrane is right here. The inner membrane, this is the matrix. This is the intermembrane space, and this is the outer membrane. It may seem really confusing because it all kind of sounds the same, like, oh, protons are going to flow from here, but protons are going to be pumped out from the matrix into the intermembrane space. Okay, so they're going to be pumped out from the matrix across the inner membrane to the intermembrane space. And then those protons are going to flow from the intermembrane space across the inner membrane into the matrix. There's no way I really remembered that. I don't know, I just remembered protons flow from across the intermembrane, across the inner membrane into the matrix, and that just stuck with me. The main concept of protons flowing is this right here. And next, we're just showing you acetyl-CoA. It's the entry point for the citric acid cycle. There's the acetyl group right here, and this is the coenzyme A. Notice in the other slide, the acetyl-CoA looked really small. It's just the CoA, you can group it like this. No one wants to draw out this whole structure, so they're just going to write CoA with the acetyl group. The end product of glycolysis, there is pyruvate plus coenzyme A, and then it's going to make acetyl-CoA with some electrons pulled off of it, of course. Okay, next, we're going to be going over the citric acid cycle. The main thing you need to take away from this is that all of these carbon bonds are being rearranged and broken down to make to have its energy slowly pulled off of it. And with its energy being slowly pulled off of it, Dr. Shen really wants you guys to see how those carbon bonds are moving throughout the cycle. Will you guys need to do this on a test or tells him where each carbon bond is in each step? No. You just need to understand the whole basis of it. So you see how it just like moves. CO2 is being pulled off. That's why you see right here, there's two CO2s, one here, one there, but then the CO2 is pulled off, and now it doesn't show up here. You do need to know the name of each intermediate: oxaloacetate, citrate, isocitrate, ketoglutarate, succinyl, succinate, fumarate, malate. One thing that people like to remember it by people use the analogy, can I keep selling sex for money, officer? It's kind of inappropriate, but I think it's what sticks in your mind, and whatever helps you study, I don't care. So yeah, can I keep selling sex for money, officer? That's the citric acid cycle, and that's how you remember all the intermediates in it. Like Dr. Shen said, carbons are flowing in and out of the citric acid cycle. They're shuffled by enzymes through different bonds in various molecules, where each bond rearrangement releases energy. Every time a bond is moved around, the cell is harvesting that energy to use it for the electron transport chain. CO2 represents the lowest usable arrangement and is discarded. So that's why you see a lot of CO2 being pulled off multiple times. And exiting carbons are replaced by incoming ones in a constant cycle, dependent on the acetyl-CoA supplies from glycolysis. The soup thing is kind of an example where you walk into a grocery store and the soup aisle is always stocked. That's because the store employees are stocking the soup constantly, and it doesn't look like anybody took the soup out to buy it, but they have been, and the employees have been stocking it up. That's the same way our bodies have been replacing those carbon bonds with new ones from our from our resources. So carbons would enter as acetyl-CoA, but as they're recycled, as they leave, they exit as CO2, and they're just replaced over and over and over and over again. Okay, so this is what you really need to remember from the citric acid cycle. You need to remember that carbon bonds are breaking and releasing energy. We're harvesting that energy, being recycled, CO2. But if you're to study like the what goes in, what goes out, where electron carriers are needed, this is where you get it from. You study this slide. So it starts off with acetyl-CoA, citric acid, ketoglutarate, succinyl-CoA, succinate, fumarate, oxalo acetate. It's missing some. You can't say, can I keep selling sex for money, officer, because it doesn't have one of the intermediates, but this is all Dr. Shen wants you to know. And each cycle, each acetyl-CoA that goes around, you make 1 ATP. 3 NAD pluses are used. 1, 2, 3. They harness H plus, and it makes 3 high energy NADH, and then there's also a FADH2 used. You need to remember 3 and 1. There's the 5 steps of aerobic respiration. This just shows you how much ATP is in each step. Oh no, how much electron carriers are in each step. So glycolysis, you're going to be using 2 NADH or 2 NAD pluses, and it's going to yield 2 NADHs. Pyruvate oxidation is also going to yield 2 NADHs. The citric acid cycle is going to make it's going to make 3 NADHs, but also going back to glycolysis, what we were showing you, the glucose is being split into 2, so then those 2 molecules are going down the same pathway at the same time, so that's why there's 1 plus 1, 1 plus 1, 3 plus 3. In the other slide it said 3 NADHs, but now we know that there's the glucose is split into 2, that's why it makes 6, and also makes 2 FADH2s. And then next it's going to feed into the electron transport chain and go through ATP synthesis. So we mentioned FAD, but we didn't really go over what it is. It's an electron carrier, flavin adenine dinucleotide. I don't think you really need to know the name that well, but it's an acceptor and carrier that is reduced, gaining electrons. FAD is turned into FADH2. It's not as high energy as NAD to NADH, but it's still used. It's going to accept electrons from the conversion of single bonds to double bonds. That's why pulling these electrons off is going to create a double bond, because those 2 carbons are going to bond together since they're not bonded to the H's. This is just a similar structure that we've seen all along. This is a nucleotide. Nucleotides come in different types of forms. This is also a nucleotide. Nucleotides are just like really nitrogen bases like that. Dr. Shen just wants you guys to know how nucleotides show up in all different shapes and forms and have different functions. Amazing. And then you can see the difference between reduction and oxidation. It's being oxidized, so it's losing its electrons. There's no H's right here. Comparing it to this, there's those H's right there. It's being oxidized, lost those. If you want to reduce it, gaining it, you put those back. And riboflavin is vitamin B12. So, this is just showing you guys the picture again, because you'll be seeing this a lot This is a good picture to study everything you need to know. I mean, like, it doesn't show you the number of CO2 and stuff, but just to get a general basis of where everything is happening and the location and what is going on. Okay, so going over electron transport system, each electron transport system is a large multi-protein complex. There is one. 2 is not shown right here because, like, I don't know, we just don't like to mention her. And then there's 3 and there's 4. 2 doesn't really churn out as much protons as 1, 3, and 4, so we just kind of leave her out. But they're going to receive the electrons from NADH and FADH2 because those are the electron carriers. Those electron carriers are going to dump their electrons right here. They're going to be pulled off, and they're going to be sent through these cytochromes. Not cytochromes, but they're going to be moved through these different complexes, and then as those electrons are pulled off, they're also pulling those protons off of those electrons. Notice how I said, like, protons and electrons are like the same thing. They come paired together. Whenever there's an electron, there's a proton. Whenever there's a proton, there's an electron. So electrons are being pulled off. You see these electrons here, and then they're taking the hydrogens off of it. They're constantly taking the hydrogens off and pumping it off to the other side. It's going to go through those different complexes. All the hydrogens are collecting up here, and then they're going to flow down here. And we're going to be explaining these complexes next. So those complexes, there are special electron carrying groups found in the electron transport system. They're called prosthetic groups. An example is like the baseball is the electron, and the glove is the prosthetic group. The glove just helps the electron, or helps the baseball player to efficiently transition the ball. These gloves, these prosthetic groups, are going to help the electron being moved throughout the electron transport system. So there's flavoproteins. They have partial FAD flavin mononucleotides. I don't think you guys really need to know FAD flavin mononucleotide, but yeah, he's just telling you how many there are. There's iron sulfur proteins. They have an iron and sulfur core attached right here to the protein cysteines. There are cytochromes. They contain a heme group with a Fe at the center. So right here, these are the cytochromes. This is a heme group. You could also have Fe and copper at the center. So like the copper would be like right here. Those are going to carry electrons too. And then it's coenzyme Q, which is right here. Another term is ubiquinone. There's no protein bound. It's just free floating. You don't really need to know the structure of these too well. I feel like you might need to know this for your homework, but for exams, But if you do, come to the review session and I will tell you. Moving on to the next slide, you can see there's the different ranges of affinity, electron affinity. Those ubiquinone and cytochrome C has higher electron affinity than H2 and FAD and NAD+. That's why, since it has higher affinity, it's going to be placed in the electron transport chain because they like to deal with electrons more. The reverse reaction drives the reduction of molecules below it on the list because NAD has a stronger dislike of hanging on to the electrons. So that's why I said these handle electrons better. This is just a picture of the affinity and it going down to the line. So it starts off with the NADH, stripping its electrons off, going through complex 1. There's also complex 2, we just don't really talk about her too much. Gonna go down to complex 3. As you keep going down, the affinity is higher and higher. Cytochrome C. And then it goes all the way down to oxygen, which is the last electron acceptor. You need to ingrain that in your head that oxygen is the last electron acceptor. Why is it introduced so at the very last step? Because oxygen has the highest affinity for electrons, that's why it is right there. Going back to the other slide, oxygen is the last, has the highest affinity. This is just an illustration of what is going on right here, but with numbers. So, next we're going to be going over ATP synthase. It's going to be using protons from the electron transport chain. The protons are going to turn the gamma subunit, which is right here, and then the turning of the gamma subunit is going to change the shape of the beta subunits right here. And those changes are going to make ADP and phosphate come together, and it's going to those turns are going to cleave them together, and it's going to make 3 ATPs per turn. So 1 proton makes 3 ATPs. 1 NADH makes 3 ATPs. 1 protons pumped due to FAD makes 2 ATPs. I don't know why it says 2. I think it should be times 2. But you guys do need to know this, the tally of ATP output from aerobic respiration. So each step shows the ATP made from it, and it shows you how it was made, substrate level phosphorylation or proton pumps. I would memorize this slide because he will definitely ask you lots of questions about each step and whether which one is substrate level phosphorylation, which one uses the proton pumps. And you need to know the electron carriers in each one that are in use and the different amounts of ATP. So the total yield of energy from one glucose molecule is roughly 38 ATP. Obviously, that's not an exact number, but science likes to group it as 38, so we're just going to go with it. This is just a fun slide to show you guys. Cyanide interferes with cytochrome C, and when the cyanide is going to bind to the cytochrome C, which is a very important electron acceptor, it's going to bind there, and now it's going to be inactive, and then you will die. 1 gram of cyanide can kill a 160-pound person within 10 minutes because you stopped the production of ATP. This might show up on your test. It's just like a 1 question thing, but this is why I want to show you guys this slide. Next, we're going over photosynthesis. Photosynthesis consists of 2 parts: capture of solar energy and conversion into chemical energy, and then using ATP to rearrange the CO2 that it harnesses to make carbohydrates. Energy transduction and carbon assimilation. So there's the H2O energy, and it makes O2, and then it takes the carbons and the ATP to make sugar. So right here, it's just like, I feel like they should have split this up. So you're going to use light and H2O to make O2, and you're going to use CO2 to make sugar. But it's all happening like, this happens first, this happens second, but the whole reaction, this is what's happening in photosynthesis. And then looking at photosynthesis, you can tell that this reaction, if you flip it around, is phosphodative oxidative phosphorylation. Oh my gosh. Okay. You're gonna flip it around, and you're gonna be using your sugar, and it's gonna be making CO2 and H2O, which is what we respirate, except the light part. So chloroplasts have photosynthetic factories. Each leaf has 20 to 10 chloroplasts per cell. You can see right here, there's the stroma, it's gel-like and filled with enzymes. There's thylakoids, flat-like sac structures right there, and then the granum is a stack of thylakoids. It looks like a stack of pancakes. Yeah, it's just showing you the structure. Photosynthetic prokaryotes don't have chloroplasts. They are like free-living chloroplasts themselves. Remember the endosymbiosis theory? We're not going to be talking about that, but yeah, it's just showing you cyanobacteria don't have chloroplasts. So this is the picture of photosynthesis, is what's going on. There's going to be the light harvesting complexes right here. First, this is energy transduction, and then there's carbon assimilation. It's kind of like oxidative phosphorylation, but backwards. You can see. There's the electron transport chain first, and then it's going to use those electrons, go to the Calvin cycle, and then make glucose. So, I wanted to go over with you guys chlorophyll A and chlorophyll B. Light is captured by specialized molecules. They're called chlorophyll. Chlorophyll B is much more present than chlorophyll A, as you can look here. Chlorophyll A is that light green color right here, and chlorophyll B is this dark green color. One thing to note about this is that we use chlorophyll B a lot more often than chlorophyll A, because chlorophyll B has more range of light that it can absorb on the spectrum rather than chlorophyll A. Those are both green because they're going to be absorbing all that spectrum of the light, but then they don't absorb that green part right there. They're going to be reflecting it. That's why chlorophyll is green. But chlorophyll B takes in more ranges of light than chlorophyll A. So, light are basically little packets of energy. Has waves and particles, so you can say like a particle of light, a photon, or waves of light like this, something you've seen pretty often. Shorter wavelengths mean higher energy, so you can see here these wavelengths are really short. This is towards the ultraviolet spectrum, and as it goes to the infrared part, the wavelengths are longer, and that's why we can use like microwaves and stuff because they're lower energy. Say you wouldn't want to use these like gamma radiation rays to heat up your food. You're gonna get like cancer because it's such high energy. Okay, so the photons are going to excite the electrons in the chlorophyll atoms. The photons are basically going to be jumping around in the chlorophyll, and then it's going to start off with the electron in its ground state, and then electrons are going to absorb the energy of the photon, and it's excited into a higher energy orbit, and they're going to be jumping around. Chlorophyll A and B only excite at certain wavelengths. The molecular structure determines its sensitivity. Like the structure of a cup depends The structure of a cup determines how much rippling of water there is to the frequency. So this is chlorophyll A, and this is chlorophyll B. There's a bigger spectrum. Okay, this is what I was talking about, bouncing around. Photosystems transfer excited electron energy. They are thylakoid membrane complexes of proteins, and chlorophyll will establish that proton gradient. Some chlorophylls act as receiving antenna. They pass energy onto other chlorophyll. The collection focuses on special chlorophyll molecules in the reaction center that pass out of that photosystem. So this is the photosystem. The photon's going to come in and it's going to be bouncing off all these different reaction centers, and then it's going to have the electron transfer and the electron acceptor. It's kind of like the kinetic balls. I guess the picture is blocked right here, but once you pull a ball up, it's going to transfer that energy to the other side. It's going to keep swinging back and forth until the energy is reduced. This just shows what is happening in these photosystems right here. This is a photosystem. That's all that's happening in there. So one thing to remember in photosynthesis, it's going to start off with photosystem 2 and then photosystem 1. Photosystem 2 has a special chlorophyll molecule, chlorophyll A molecule, that is sensitive to 680 nanometer wavelength. So it's P680 right here. First you're going to have the photon come in, and it's going to bounce around all the reaction centers, and then it's going to be passed on to P680. And then that P680 is going to transfer the electrons to plastic quinone, not ubiquinone, but plastic quinone. It all kind of sounds pretty similar, but you need to remember it. Plastic quinone is shown as this, QB, and it's going to bind to the H plus. The H plus is right here, and it's going to turn into QBH2 right here. And then the missing electrons from chlorophyll P680 is replaced by breaking more H2O and O2, and the H plus is going to diffuse out. Wait, you're going to have the H2O and O2 right here, but the oxygen is going to diffuse out of the cell and is released into the air as waste. It's going to be taking those H pluses. So this is the waste that's coming out. And then the QBH2 is going to take the additional H pluses to cytochrome BF, which is going to transfer those H's to the lumen right here. There's a thylakoid space, thylakoid membrane, stroma. The lumen is on the inside, and then the result of all those protons building up in the gradient is going to fuel ATP synthesis. This is the molecular structure of a photosystem. You don't need to draw any of this, but he just wants you to see how all of the photons are going to be bouncing off of those reaction centers, and they're going to be going through the photosystem, and it's shown right here, those light harvesting centers. Okay, and that is it.